Dear students, may Allah protect us all. In this pandemic of COVID-19, as the university is closed, the faculty members uploaded their relevant subject course materials on the university website. I did the same. Then to bring you on the track, I engaged you on WhatsApp. For that, I divided you in two classes, WhatsApp boys class and WhatsApp girls class. And I appreciate both the groups. Actively participated in topics discussion. Then upon the request and insist of you guys, I started sending you in WhatsApp group my recorded lectures. That was continued till 13th recorded lecture, and we discussed topics, and we discussed and we also discussed the question answers, and we had many things in those recorded lectures with you people in WhatsApp groups. Again, it was also you people's suggestion to start it on YouTube. So apart from the 13 classes on WhatsApp, I have uploaded 15 recorded lectures on YouTube. In this way, total 28 classes so far, and almost we are near to complete our course. And in that YouTube classes also, students asked any question in WhatsApp group or commented on the YouTube. So I appreciate your efforts, and again we can pray that may Allah protect us all in this pandemic of COVID-19. So here I have summarized the things for you, and to bring the things once again in your mind, I have summarized what we have done so far. So let's start here. Introduction of the subject. We define the subject, what is epidemiology, and then we learned that what is epidemic, then different definition of the epidemics. Then we briefly introduced to the subject that what the subject is related in, and finally we defined the subject as the science that studies when and where diseases occur and how they are transmitted in population is called epidemiology. Then we talked about the purpose of the epidemiology. Then we little about discussed the uses of epidemiology, and then we discussed in a little bit detail about the importance of the subject. So we covered these things: the introduction, the definition, and the importance of the subject. These are the lecture notes provided to you, and which are available on the university website. So you can go through these notes which I have described: the use of epidemiology study, importance of the subject. Then we started the main branches of epidemiology, like the first we studied and learned about the descriptive epidemiology. Then in descriptive epidemiology, we studied the essential characteristics of disease that we look for in descriptive studies: place, person, time. And then we also discussed the variables and the variables of the essential characteristics, like the person variables, place variable, and time variables. Then we talked about the descriptive epidemiologic studies, in which we studies case report study, case series studies, and cross-sectional studies. These are the main three types of the descriptive epidemiology. I have given you one example, that of the chronic sleep loss of people in the United States regarding a survey in 2006. And that was an example of the cross-sectional studies. And here we learned that what is hypothesis. So we learned here that hypothesis is an educated guess that is testable in in a scientific investigation. So descriptive data, we discussed that who, where, when provide information to develop hypothesis. So we summarize it in descriptive epidemiology, that descriptive type studies and descriptive epidemiology generate hypotheses or develop hypotheses, and these are the examples of the hypotheses which we discussed in during the lecture. Similarly, then we moved next to the analytical epidemiology. We defined analytical epidemiology. We discussed the essential characteristics of analytical epidemiology, that was host, agent, and environment. Then we discussed a little about the variables of these three essential characteristics, and we also studied that epidemics occur in host, agent, and environmental factors are not in balance. Then we started the study types of the analytical epidemiology, in which we started the cohort studies, and then we discussed about the case control studies. So these were all about the things so far we did. Then we started to understand some basics of some basic definition of things which are coming in our way many times in the subject. So we learned those things first. For example, we learned what is the definition of infection, what is a disease, and then types of infection like local infection, systemic infection, focal infection, primary infection, secondary infection, subclinical inapparent infection, then sepsis, septicemia, bacteremia, toxemia, viremia, and all these terms are coming again and again in our different lectures recorded here. So these were the terms or things we learned. Next we also discussed about that what is sporadic disease, what is endemic disease, epidemic disease, what is pandemic disease, acute disease, chronic disease, subacute disease, latent disease. We discussed all these things in very detail in some lecture. Then we discussed that what is the incidence of a disease, what is the prevalence of a disease, and I told you that we will discuss these things in detail later on. So far I have in this summary, I'm not talking about that next chapter which will be discussed inshallah in our next coming lectures. Then we discussed the reservoirs of infection. A reservoir, we defined that a reservoir may be living or non-living, and this is a source which provide the adequate condition for survival and multiplication, opportunity for the pathogen, and also opportunity for the transmission of the pathogen. So that source may be living or non-living. We discussed in detail that that's the reservoir may be human, animal, which are the living part, living reservoirs of infection, and then the non-living reservoirs of infection. Then we discussed the human reservoirs of infection in detail with examples. We also discussed the animal reservoirs in detail with examples. In animal reservoirs, we discussed about the zoonosis. In the non-living reservoirs, which are the main non-living reservoirs are soil and water. So we discussed about the soil and water. Then we started routes of transmission, and in routes of transmission we discussed that the three principal routes of transmission: contact, vehicle, vectors, then Contact transmission has further 3 types we discussed: direct contact, indirect contact, and droplet transmission, which is called droplet nuclear transmission. And each of the type of contact transmission, I have given you the examples in a little bit detail. So these are things we studied. The next was the vehicle transmission in routes. Amongst the routes of transmission, we studied the vehicle transmission, the transmission by a medium such as water, food, or air. In detail, we discussed that what is waterborne transmission, what is foodborne transmission, and what is airborne transmission. We also discussed about the vector-borne transmission or vector transmission or propelled vectors, and then we discussed the mechanical transmission by a propelled vector and biological transmission. So mechanical transmission is the passive transport of pathogens, and biological transmission is an active process and is more complex as compared to the mechanical transmission of the vectors. So these were the things we discussed. In the immunization or vaccines topic, we learned about the immunity, that 2 types of immunity: adaptive immunity, innate immunity. Then further have 2 types, natural, artificial, and then we discussed all these things. So in this topic, we a little bit went to the vaccines in a hurry. That vaccine is suspension of organisms or fraction of organisms that is used to induce immunity. And what is herd immunity? These were the things we discussed. Then we talked about the types of vaccines and their characteristics, in which we discussed about live attenuated vaccines, inactivated killed vaccines, subunit vaccines, toxoid vaccines. All these things we discussed in detail there in the lectures. We discussed about the nucleic acid vaccines. Then in detail we discussed about the future prospects of vaccines, and also we discussed about the development of new vaccines and safety regarding vaccines, people concerns about vaccines. We discussed all these things in this topic. Amongst the life-threatening diseases, the lectures were included with only 2 diseases, which were AIDS and hepatitis. So we discussed in detail about the hepatitis in AIDS. Here is the slides which we talked about, which we discussed regarding the AIDS, the structure of the virus, genome of the virus, HIV clades, HIV1, HIV2, HIV infection. Here the most important thing we talked about is the one of the most common misconception is that HIV infection is synonymous with AIDS, but this is not the case. AIDS denotes only the final stage of a long infection of HIV. So then we discussed about the infectiveness and pathogenicity of HIV. We went to the different stages of HIV infection: phase 1, phase 2. These are the clinical phases, and phase 3, which is the last phase in the disease, and it is called a clinical at this time at phase 3 we call the HIV infection as AIDS, because this is here in phase 3 the clinical AIDS emerges. Resistance to HIV, survival with HIV, diagnostic methods of HIV, and HIV transmission, routes of transmission of HIV. We discussed all these things in detail. Then we discussed about that AIDS worldwide situation or status of the AIDS worldwide. Then AIDS in Pakistan we discussed in detail. And then preventing and treatment of the AIDS we discussed. These were the things we discussed in the last in the recorded lectures. Then we started hepatitis, and we amongst the 5 viral hepatitis: hepatitis A, hepatitis B, hepatitis C, hepatitis B, hepatitis E. we talked about only two hepatitis, hepatitis B and hepatitis C. These both are, B and C are life-threatening hepatitis. We discussed in detail about these hepatitis B and hepatitis C. These were the slides which I shown to you during the lecture, online lecture delivery. I am going through just to have a vision of you people in a summary form. Hepatitis C, we started in hepatitis C. We talked about the genome, then the prevalence and worldwide status, and then we talked about the prevention. Then we talked that there are some other viruses which are still to be known better. Among those is one is hepatitis F and hepatitis G viruses. So these were all the things we discussed, and I summarized these things. So